Thanks. I'll get started. So I'd like to thank the organizers for inviting me here, and I'm very pleased to be at the single-cell meeting to hear all these single-cell talks. I should say that I myself am very new to the single-cell field, but it's great that I get invited and, irrespective of this, present some of our data. So I don't have 40 minutes to fill with single-cell slides, but probably about 30, and then I'll just show you something else, the other things we're doing in our lab. So I'm a senior scientist. I'm at EMBO for nearly 10 years. So at the end of this month, it'll be my 10th anniversary. And I will speak about largely single-cell and big data approaches to study genomic variation. We've heard a number of very good talks this morning about how one can measure DNA in single cells and how one can relate it to other readouts, such as transcriptomes, for instance. And my laboratory is really a genetic variation lab. We're trying to understand how genetic variation emerges in the genome, how it is selected for, and so what are the determinants behind its formation and selection, and largely distinguishing two classes of genetic variation, single nucleotide variants and larger structure variants, which you heard today are very tricky to call in single cells, although approaches are clearly getting better and are, that's exciting to see, are currently emerging. So we'll soon be able to tackle this form of variation, presumably also fairly good. Right now it's, apart from copy number, of course, quite tricky to detect it. But we saw today certain ways to get closer at structure variation in single cells. We study these not only in single cells, but also in bulk, and we relate them to various diseases. Indeed, lots of studies have shown recently that structure variations can be responsible for a range of phenotypes. So typically in a genome there will be many more SNVs and whether you are in a cancer genome or in a germline genome, but the structure variations are so large that they can confer large effect sizes and hence have been implicated in a number of diseases. And in cancer they are sometimes the most abundant form of variation. Actually, a number of cancers that is certainly true. So we've been for a long time also interested in developing methodologies to detect this form of variation in the genome. I'm showing here a very old slide that I devised before I started EMBO. That's from my postdoctoral times at Yale, where I co-developed, this is one of the product of our postdoctoral research, I co-developed an analytical method that together with an experimental approach could allow the detection of structure variations in bulk sequencing that has been very widely used since, and in different flavors, not necessarily in the flavor in which we developed it. We used an ancient technique called 454 sequencing at that time to shear genomic fragments, circularize them so we can capture the ends of them and then map them to a reference genome. And nowadays that principle has stayed. Even the principle of circularization is still around with various flavors, nowadays known as made-per sequencing, and which the principle that has stayed is the size selection and relating the sizes to a reference to infer structure variations. So this method obviously has been widely used. This is certainly my highest cited first author paper, and will probably remain that because I'm a PI now. It has become certainly standard in genetics and genomics, so this is how people currently detect structure variation, unless they're happy with going for copy number. However, it has key limitations too, and I want to be really upfront with them. So we need to detect the structure variations. We need to be able to bridge the breakpoints of the variants with reads or read pairs. And several talks today alluded to this. So in single cells, if you're missing coverage, it will be extremely hard to identify the structure variant, or at least to sensitively call it, because each time you're losing a read that reaches over the breakpoint of it, you're losing the variant. This limitation comes with yet another limitation, not only for single-cell sequencing, but for bulk sequencing. These methods break down once the structure variant breakpoint is in a repetitive DNA piece. So, and whether you're in the germline or in cancer, you'll often see actually that breakpoints are in repetitive DNA. So telomeres can undergo rearrangements, for instance. Centromeres can undergo rearrangements. In the germline, you'll see lots of larger repeats in the genome known as interspersed repeats or segmented duplications that facilitate the rearrangement of sequence and hence are at the breakpoints of SVs. And then these methods unfortunately don't work. And that's specifically pronounced for one form of variation in the genome known as an inversion, which in the germline is nearly always flanked by long repeats at the sites. This comes from the fact that inversions typically form in the genome by recombination, at least the large ones. And nearly all of the ones greater than a few tens of kilobases formed by that mechanism. So you have long repeats in the genome that anneal and undergo unequal crossover, a non-allelic or homologous recombination. And hence the intervening sequence can then flip in orientation after this crossover has taken place, and leading to an inversion that with current sequencing techniques is hard to trace or not traceable, because if this repeat is long, we will not be able to reach over it. In reality, there's a number of long inversions in our genome that are framed by segmented duplications that are several hundreds of kilobases in size, sometimes a megabase in size. So even with a long PacBio or nanopore read, you will not necessarily be able to penetrate it and hence miss this form of variation. So these inversions have been, until recently, assumed to be unsequenceable. And I recently became very interested in seeing a new technology emerge as we are indeed interested to detect all forms of variation in the genome that actually allows to, that has the capability to detect inversions. This technique sequences individual strands of the genome and also sequences genomes in a homolog resolved manner. I'm calling it here a new lens in next generation sequencing. Certainly, I think this is quite powerful for understanding DNA variation, as I'll hopefully demonstrate in this talk here, because it can not only show us inversions, but also can completely haplotype resolve genomes. And we can integrate that with other data types to then get genetic information for a diploid genome fairly precisely mapped. So we all know, of course, this is fundamental information known since many decades, that the human genome is diploid and that the DNA has two complementary strands. However, sequencing methods chop the DNA and lose the information of which homolog a DNA piece emerged from and also which strand it emerged from. So we're not really considering the Watson and Crick strands of the diploid genome as we sequence. We can try to reconstruct the information after sequencing, but of course it would be much better to detect homolog state and sequence strand as we sequence. And that is actually, since a few years, possible, since work that was led by Esther Falcona, Ashley Sanders, who's now a postdoc in my laboratory, and Peter Lansdorp. They developed a technique called Strand Seek that allows to sequence the DNA for individual strands. It was initially developed to measure exchanges between identical chromatids, between sister chromatids in G2 phase, but what recently emerged is that there's actually other uses for this technique to detect haplotypes and inversions. And so this technique works by introducing, during replication, so in a mitotic cell, a label to the replicating strand. So when DNA replication starts, the DNA obviously unwinds, and this is indicated here for two homologs of a given chromosome. And then replication is initiated, and in Strand Seek we perform replication given the cells a drug that's called bromodioxyuridine, or BRDU, which will readily incorporate into the strands, and at the end of replication we'll hence have a 4-end cell in which one strand of each chromosome is labeled by BRDU. The BRDU labels will, on average, be present every 100th to 150th base. It's fairly dense labeling. The way that replication works, replication is very exact in that. You start off with an intact DNA strand, and then you use one strand as a template, and you fill in the other side. The other strands also used as a template and fill in on the other side to create 4N from 2N, which implies that here we really have chromosomes where across the entire chromosome one DNA strand is not labeled and the other is labeled. As for useful information, we do not yet stop for library preparation here, but we let mitosis carry on what's happening now is a random segregation of these two pairs of sister chromatids. So that's a very important concept for Strand Seek. The cell during mitosis will very faithfully ensure that each homolog will move at least once and exactly once to the daughter. So there will be a paternal and maternal homolog, P and M for father and mother. However, the cells will not control whether it's this one or that one. So cells do not have checks in place for which strand acted as a template. That's random. And this randomness actually turns out to be useful for Strand Seek because we'll have, in every second cell, the two homologs of a chromosome will be sequenced on a different strand. And we can obviously make use of this information. We do that by using micrococcal nuclease to digest the genomes, and then ligate Illumina adapters that are barcoded with a directional barcode, so that we can later, we can keep the sequence orientation information, and then we can later remove the BRDU containing strands completely by nicking and performing then a linear amplification. And what we end up having is, so this is also a WGA-free methodology, but it's not amplification-free, so it's probably also pre-amplification-free. So we amplify at this stage here when we have a purely single-stranded library. We bring this to higher depth. And then we sequence the genomes with approximately 0.1 to 0.15x coverage to generate sparse genomic information for which we can reconstruct inversions and haplotypes. This is shown here. So for inversions, it's actually fairly trivial to identify this form of variation in Strand Seek because an inversion implies that sequence reads just simply map to the other strand of the genome. So we have here a reference in which a segment is inverted relative to the sample. And since we will sequence the sample on just one strand, once we map back to the reference, anything that maps to the wrong direction will immediately tell us an inversion has happened. And this is a single cell with, in this case, quite a bumpy coverage. So we're now getting better than the coverage shown here, in which the inversion lights up quite strongly. So it's a fairly long inversion event. This one is one of the larger inversions in the human genome, a 5 megabase polymorphic inversion present in a large portion of the population. However, it's not sequenceable by standard methodologies because it has very long repeats that frame it. So currently, people would use fluorescence in situ hybridization, FISH, to detect it. But there's a second trick we can apply, and that's quite important one once we integrate this methodology with other methods. And that is each time the two homologs of a chromosome pair are sequenced on different strands, we can start to sort the reads onto the different haplotypes. And here's indicated, so this would be a cell where these reads map to the genome, and we'll have, for one chromosome, reads mapping in forward and backward orientation, and can from that reconstruct the haplotype, because as soon as a chromosome is sequenced on two strands, we know that one homolog has been the one strand and the other has been the other strand. And we've computed how long the haplotype blocks get. Well, not surprisingly, but reassuringly, we get haplotype blocks that comprise the entire chromosome. And that is because this, again, replication uses really exactly one template for the entire molecule. And it's not, so this method is not, as other methods, confounded by centromeres or long repeat regions. So we get a single haplotype block. Irrespective of this, the data is sparse. So we need to get really dense haplotypes, not just long blocks. We need to integrate with other methodologies. Before I show you how we integrate, this is another view of how Strand Seek data typically looks like. I hope people in the back can readily see what's happening here. So we show essentially a single Strand Seek library worth of data plotted onto chromosomes. Hope that's not your kid. I want to speak to you. I see, it wasn't yours. So this is how reads mapping for a strand seek library look like. And since for every chromosome we have, again, random segregation, some chromosomes will show up just in one color, like this one here. Chromosome 7 will be sequenced on the Watson strand only. In this case, both paternal and maternal homolog were sequenced on the same strand. But then there's others where we see sequencing happening on different strands. And we see here, this is, in this case, a male sample. When we integrate this information with SNPs that we collect, we can readily integrate the data across haplotypes and generate from that a haplotype map. So we can not only separate these signals for individual chromosomal homologs, but also use this data to better feed into haplotype inference. So together with Tobias Marshall's group in Saarbrücken, we developed a method to do this. This is largely Tobias Marshall's group's work, I should say, in this case, an algorithm that uses the read data, which are again sparse, and walks along the genome and takes all haplotype informative reads to build up haplotype scaffolds that can then be integrated with other data. So if we, for instance, Illumina sequence is sample 2, we already get better haplotypes than with single-cell sequencing alone. But we can also use other technologies, such as Pacific Bioscience sequencing or 10x genomics, a technology that uses so-called linked read sequencing to get very dense, low-range haplotypes. So it's quite complementary to Strand Seek. It doesn't give you long-range haplotypes, but small-range haplotypes that are dense and can, once integrated with Strand Seek, lead you to phase nearly the entire chromosome. How this scales with the number of Strand Seek cells is shown on this slide here. So we are considering 4 different techniques here, or 4 different combinations of techniques, I should say. This is Strand Seek plus Illumina sequencing, Strand Seek plus PacBio sequencing, Strand Seek plus 10x genomics, and 10x genomics plus PacBio sequencing. And we are, in this case, coloring the haplotype density and haplotype switches using different colors. So each time it looks like a salami, that's actually bad, because the haplotype information is lost over a certain region. And 10x genomics, which is frequently used for haplotyping, again, gives you dense haplotypes, but they're relatively short. So then the information is lost. However, just integrating it with 5 Strand Seek cells will nearly guarantee that along the entire genome you'll have fairly complete haplotypes. And with increasing the number of cells, this of course gets better. One can also integrate Strand Seek with PacBio sequencing, generating not quite as good, but still reasonably looking whole chromosome haplotypes. So with this, we went back to as I'm co-leading a structure variation analysis consortium since actually the 1,000 genomes times. And so one of the questions that has really has become more a side question in recent years, but has guided some of our researchers performing better methods development, detecting variation in the genome. So this is an old slide from 2015 where I reported on our results from sequencing 2,500 genomes by 8-fold coverage sequencing. That's not bad, but fairly low coverage for bulk sequencing. And we've identified about 4,000 structure variations in per person in 26 different populations. And we were quite interested to go back to this data now with this capabilities of Strand Seek to ask the question, to what extent do the long-range haplotypes that Strand Seek give us yield us a better picture of genetic variation that we can see in an individual genome. And within the consortium, we had already a while ago begun sequencing genomes with a range of different technologies, including long reads, initially PacBio, now also Oxford Nanopore. We've been using 10x genomics for dense but short-range phasing, as well as bio-nano-optical mapping, an optical mapping technique that also gives you insights in structure variation and Illumina sequencing. And we've integrated all of these technologies to generate a haplotype resolved, nearly fully haplotype resolved map of a structure variation in initially 3 family trios, so 9 individuals overall, where we could use the trio design. So trio means mother, father, and child. We could use, since haplotypes tend to be inherited, at least partially, we could use the trio design to assess to what extent we got the haplotype information right. And so this is here a slide showing the length of the blocks that we generate, the haplotype blocks, which we generated for SNV phasing. Overall, we generate a call set of 27,000 structure variations, greater than 50 base pairs per human genome, versus 4,000 seen in the 1,000 Genomes project. But of course, there are other studies nowadays using long reads, which get fairly much closer to this number than the 4,000. So this is the historical gain. It doesn't reflect the gain that one would achieve from sequencing PAC biosequencing, from performing PAC biosequencing alone. However, there's quite a gain in sensitivity and specificity at the individual call range for getting the haplotype information right. We, of course, were interested largely, since we are looking at Strand Seek so closely, to assess to what extent we can learn more about inversions in the genome with this technique. And we hence employed Strand Seek to locate inversions separately from the multi-platform integration effort in these individual genomes. And one can see here how this would go for a putative trio, for instance. So we assess individual genomes, and at a trio level, the status of various regions in the genome. So a heterozygous inversion will imply that one of the two strands flips in orientation. And this can, depending on whether the two homologs of a given cell were sequenced on two strands or on one, will look differently. So here, most of the chromosome was sequenced just on one strand, and one of these flips. And in this case, just in a different cell, both chromosomes are sequenced on different strands, and just one strand flips, generating the same signal. And integrating this data across cells gives us the inversion back. For homozygous inversion, it will look like this. You get a full flip of sequence as both haplotypes are affected. When we generate an inversion map in these individuals, we identify 160 inversions per sample. That's 4 times more sites per genome than prior studies. And it is a nearly 500-fold increase in inverted bases compared to the 1,000 Genomes project. Again, not quite as much when we compare to PacBio and Oxford Nanopore, but still significantly so. As only with Strand Seek we actually see the very long inversions that are present in the genome, flanked by repeats, because with this technique we're not hurt by the fact that flanking repeats harm the read mapping and hence inference of the variant. So we're comparing here different technologies, jumping Illumina libraries, that's BioNano mapping, Illumina sequencing, PacBio sequencing. As you can see here, the majority of inversion calls that we generate in this project came from Strand Seek. And what's more, all of these inversions that are larger than approximately 5 kb in size, even if not initially called with Strand Seek, could be verified by the technology. So all in all, we now have a call set about 500 inversions, which doesn't sound tremendously large, but this variant class was quite underrepresented in databases. It's so hard to map. And we went back to the literature to assess, can we learn something from the way these inversions are distributed in the genome? And we tested for intersection between inversions and critical regions of microdeletion syndromes, because there was prior literature showing that for some of these microdeletion syndromes, actually 7 already known ones, long inversions are present at the risk locus. So what is a microdeletion syndrome? So it's a syndrome that occurs through de novo mutation of a microdeletion or microduplication of hundreds of kilobases up to over megabase in size. And they affect regions in the genome that are more frequently rearranging than normal regions would be. So there's certain critical regions for these microdeletion syndromes that, which explain their frequency and abundance, and and classification into common phenotypes. What we found is, so seven instances of long inversions flanking microdeletion syndromes were previously known. We've identified 58, so we are finding quite a number of additional such instances actually. So it seems to be more common, more the norm than an exception, that long inversions are present at the breakpoints or within microdeletion syndromes. I'm showing here as an example a critical region for 3q27 microdeletion syndrome, which comes with a very specific clinical phenotype and a developmental delay, and in this case identify an inversion sitting right at one of the breakpoints of this common microdeletion event, and as you can see here, encompassing segmented duplications that are thought to mediate this microdeletion risk. So that would imply, if true, that would imply that the inversion status would be actually a risk factor for whether the microdeletion occurs or not, because it would flip the sec tube and bring it to a new location, and afterwards it might not be able, or would not be able, to recombine anymore in the same way, causing the microdeletion. As I said, 58 examples were found overall. There's some quite interesting ones where we found several inversions marking different previously reported microdeletions. This is from a catalog of microdeletions. This is the critical region, which has been generated from the catalog, but one can see that there's different inversions that appear to map to different subsets of these deletions. This includes a case of two flanking microdeletion syndromes on chromosome 16p, where we identified altogether 3 inversions marking 3 of the segmented duplications involved. Having said that, we don't yet know whether these inversions act as risk factors, but given that they overlap with the breakpoints of these regions, and particularly with the segmented duplications, that is at least not unlikely. All right, so I've used up nearly 30 minutes, but as I predicted, I'm now at the end of my single cell talk, and I would like to spend the last 10 minutes or so to tell you about something else. But I'd be happy, if there are any questions, I'd be happy to answer them now around the single cell work. If not, I can, of course, immediately go on. Yeah?
So knowing these microdeletions and inversions, can you go back to the standard sequencing data to figure out why your algorithms missed them, or is there something, or they just not, is it something about the standard sequencing that just, does not allow you to detect them in the first place, or is it just less sophistication in these algorithms?
Yeah, that's a good one, a very reasonable question. So sometimes we can go back to the data and identify that there's this, there will be occasionally one read that's long enough from a long read technology to pass through, but then the algorithm will not have called it. We'll have seen just one read. We do, interestingly, even for bio-nano mapping, we find fairly poor concordance. So bio-nano doesn't give us these long events. In most cases, when we see these inversions, we immediately realize why they haven't been called. So they are mediated by very long repeats, hundreds of kilobases in size. So I guess ONT needs to advance further for us to get them to direct sequencing. Yeah?
Is it clear to you maybe how this mechanism, or how these mechanisms relate to high-level amplifications of oncogenes under therapy, like B2 or EGFR or whatever? So
how
any investigations by anyone in that direction?
You mean how basically this single cell technology relates to that?
No, the microdeletions and inversions and so on.
Ah.
And what you said about the role of repetitive regions.
I think that these diseases are all relatively rare, so that interestingly, some of these diseases are associated with cancer or with overgrowth symptoms. In these cases, though, probably the patient populations will be so small that, at least I'm not aware of any systematic studies yet with respect to amplicon formation that is specific to a microdeleted locus.
Maybe not a microdeleted locus, but in general, let's say how your technology could help to solve these cases of, let's say, what has happened to in the genomes of these cancer patients.
Well, I mean that to me is kind of a different, or it pertains to a different question. So the question is, can we use Strand Seek to study cancer? And that's an interesting one. So we're looking into this currently. Also in cancer, you get large inversions in the genome. The necessity to haplotype reserve what's going on will also help you. So I think there's something in there, but yeah.
So, just to take Benson, so are these inversions frequent in the human population, or are they relatively rare?
So there's we find about 160 to 170 alleles per person. So they are frequent. They also contain quite a number of genes. To what extent they are causing strong phenotypes, that's not known, and there might not many of them might cause a strong phenotype. What they will do though is they'll rearrange quite sizable fractions of the genome, and by doing that, kind of change the context of the genome so that new rearrangements become less or more likely, which I think is what's happening in these microdeletion syndromes. You could, of course, assume maybe because the genome changes its structure, also cancer-associated alterations may be more or less likely, but that we don't know yet.
Right.
Yeah.
I was actually asking you, so if you have like the 17q21 inversion, which is really common in the human population, like 35% in Europeans. So the ones that you found, the new ones, are they show more or less the same frequency in the human population? You found any really widespread inversion, or all of them are just happening in like 1 or 2 individuals?
So we, quite interestingly, they're often fairly abundant actually, and so what we seem to be seeing here is quite some potential for reformation of these events. So it's I guess it's a bit early days, but we've begun to look into different families from different populations, and what do we occasionally see as similar inversions, different haplotype contexts, which implies that these inversions they recur. So not only do they trigger potentially other events, but they themselves flip back and forth. And that again has been described for a few loci, but we seem to be seeing it's more the norm than the exception, that this flipping. So as of course, it's quite interesting to think about this, because that would be quite a confounder to human genetics in these regions where we see these rearrangements occur, because they'll essentially change linkage blocks. They'll probably change the way the regular environment is interpreted to some extent. Yeah? Maybe not largely, but these could be subtle things that perpetuate into genetic analysis, or they could be less subtle. And to study that will be tricky. We need larger maps of these events, but it's certainly interesting. Further questions? No more on inversions. So I now have 5 minutes left, so I have to fly through the remaining part. Which is good though, because this is not necessarily single cell related. So I just want to take the opportunity for 5 minutes to tell you what else we're doing in the lab and what else we have been doing for quite some years. So we've been assessing genetic variation not only in normal genomes, but also in the context of cancer. And reasoned some years ago there would be quite some potential to capture genomes that have been generated in different projects within the International Cancer Genome Consortium, put them together to increase cohort sizes and essentially accelerate studies of cancer evolution and linking of genotype and phenotype. And we reasoned that to do this, we need to actually become better in sharing data and generating unified and standardized variant calls. And when we initiated a project some years ago that seemed relatively straightforward, but required us to massively share and integrate data across sites to get there. And so this is the principle of pan-cancer analysis. And Since I have only 5 minutes, I'm probably going to be almost stuck with the motivation why we wanted to do that, rather than giving you results, but that's probably fine, because I wasn't really invited for the pan-cancer analysis anyway. So I'll show you more why we believe it's intriguing to perform these analyses. Well, there's quite some hidden information in cancer genomes that one can only tackle with getting large data sets, and that particularly pertains to what's happening in intergenic regions. Genetic variation obviously is not confined, in cancer is not confined to coding regions, but occurs throughout the genome. And cohort sizes, though, to tackle variation in intergenic regions need to be much larger than in coding regions, because we have a poorer understanding of intergenic regions. They're larger. 99% of the genome is intergenic, and the position of regulatory elements that are being used by genes differ from tissue type to tissue type. So quite an interesting and difficult problem that we began to tackle some years ago by assessing to what extent are alterations that are intergenic possibly contributing to cancer. And we decided to do that by looking for structural variations, for us the most favorite form of variation in the genome, and interpreting the genome in its 3D folded space that we know to exist since excellent work from the Mundlos group and Rick Young's group over the past years, and Bing Rand's group. So we know that the genome is folded into topologically associating domains, and that gene regulation largely goes on within these so-called TADs, but that there is essentially no or very few regulatory contexts outside of the boundaries of these TADs or topological association domains. So they presumably insulate, and we hypothesized that structural variations that are intergenic may actually disrupt these, or possibly even create new ones, hence lead to a potential for gene dysregulation that may be unexplored. And to explore that, we developed a computational algorithm that we termed SESAM for cis expression structural alteration mapping, which looks for structural variations indicated here that cross the boundaries of TADs and assess in this context gene overexpression. So in other words, we looked within the genome for instances where rearrangements affect the regulatory genome, and measure the degree to which they possibly affected the regulatory genome by looking for dysregulation that associates with the structure variant. And the algorithmics behind that are fairly similar. They're actually borrowed from eQTL mapping, but are considerably refined, because we have here a somatic variant class and are refined for all kinds of confounders that could occur in a pan-cancer analysis. But again, it's a similar principle. And applying this principle using the TCGA data set version from probably 2 years ago, we've identified 17 gene loci that undergo, based on our prediction, TAD rearrangements with high overexpression. So 17 might not tell you that much as in terms of number, but that's approximately the number of high confidence fusion genes described in these entities that are occurring at a similar frequency. So this implies that fusion genes in cancer, which are well known and well studied, they're actually fairly simple to recognize, because they create a fusion transcript that one can measure, are presumably paralleled by a different form that's in a sense similar, underlies rearrangements, but doesn't create a gene fusion, but a rearrangement of a TAD in such a way that gene regulation is that gene expression is dysregulated. And well, to convert this into publishable work, we realized we actually need to study at least 1 or 2 loci in depth, because how on earth is someone going to believe us from a purely correlative analysis that the rearrangement indeed mechanistically leads to overexpression? And we've done so in 2 loci. One of these, actually my favorite one, is IGF-2, a known oncogene in colorectal cancer that was noted to often be duplicated with a copy increase of 1, and sometimes there's a deletion right next to it. And irrespective of whether there's a copy increase by 1 copy or a nearby deletion, there's fairly high gene overexpression. And actually, the level of gene overexpression is not really compatible with the view that you simply increase dosage. It's actually way too high. We performed chromatin immunoprecipitation followed by sequencing to study this location, and found that there's actually a fairly strong, presumably active enhancer in colorectal cancer cells, a super enhancer, which sits close to IGF-2, but on the other side of an insulating TAT boundary. So there presumably will be no interaction, no means by which the super enhancer could activate IGF-2. So these duplications here, interestingly, always, when we see overexpression, contain not only IGF-2 but also the super enhancer. And they're actually tandem duplications. And so what our model that resulted from this says is that these tandem duplications, by spanning from IGF-2 into the new TAT and into the distal super enhancer, will create a neo-TAT which has a copy of IGF-2 and a copy of the super enhancer, which would then be presumably able to interact. And this is actually a testable hypothesis because there's now techniques such as chromosome conformation capture sequencing, which we use to verify that indeed, in the context of a tandem duplication, get very strong interaction, formation of a new TAT or neo-TAT, and IGF-2 overexpression. And just wondering whether I should close. Probably should close fairly soon. I'll give you the summary of this result too. So here the conclusion is we change from a wild type TAT structure with an insulating boundary that prevents IGF-2 from being regulated by a nearby super enhancer, which is clearly a vulnerability in colorectal cells. And if we create a duplication from here to there, then the result of this will be a neo-TAT that creates a perfect storm for cancer, bringing a strong super enhancer near a strong oncogene, leading to overexpression. And this is the work which recently got reviewed by others. And with this, I'd like to thank you for your attention. I think I close now because I'm way over my time. Or? I could tell you more, but...
Can I ask a question? I wanted to ask, how often do you think this is... how often do you think this occurs in cancer? Like, because it seems, okay, you have found this example. And
yes, yeah. We tried. I said this very briefly. So in this study, we tried to relate this to the frequency of fusion genes, and that's not. It's not so easy to make the comparison because a fusion gene, in principle, you could detect based on a single sample by just seeing gene A fuse with gene B. With this technique, we cannot make singlish observations. We need to identify correlation between structure variations that change TATs and gene overexpression. So what we did to correct for that, we said, okay, let's look at fusion genes that occur in a fairly large number of samples. So we said, well, if a fusion gene occurs in 10% of a sample of a certain tumor type, it's probably a common driver. Well, we should be able to detect enhancer hijacking events that are common drivers at 10% frequency within the patient population. And when we did the math this way, then we found there's as many fusion genes as enhancer hijacking events that are mediated by this mechanism. Well, not always neoTATs, sometimes it's just removal of an insulator, presumably, but by such a mechanism.
do you have any other questions? Okay, so I guess we exhausted all our single cell questions before.
Fair enough.
Let's